We've looked at glycolysis. After glycolysis, of course, we've also looked at the Rappaport Lubering pathway. We've looked at the other pathway was the Krebs cycle. Then from Krebs cycle, we looked at gluconeogenesis, and in gluconeogenesis, we saw that we have two other small pathways. That is the Cori cycle, as well as the glucose alanine cycle. So today I want us to look at glycogenolysis. I want us to look at glycogenolysis, and maybe we can also look at fructose and then galactose metabolism. Just from the word glycogenolysis, breaking down glycogen into individual components of glucose, because we know that glycogen is a storage form of glucose. And so in cases where glucose on its own is depleted, then we need to break down this storage form so that it gives us the individual molecules for which it was made. So that is what glycogenolysis is. And now for us to be able to understand glycogenolysis, we need to understand, first of all, how does this molecule glycogen look like? Much as we covered it in structural biochemistry, I think it's important for us just to have a bit of a review of how that molecule looks like, so that then when you're breaking it down, how we understand how then you're going from glycogen, the more complex molecule, to individual molecules that are glucose. But then we need to also understand, or rather remind ourselves, what this glycogen is. Okay? We need to remind ourselves what glycogen is. And of course, we've already noted that it is a storage form of glucose. And how is it stored? How is it stored? Now, if you remember, linear glucose has 6 carbons. 1, 2, 3, 4, 5, 6 carbons, like this. But this ultimately becomes cyclic, like that. So this is how a glucose molecule looks like. With 6 carbons, this carbon on the right, stream right being the first one, and that is how they are numbered. We've understood that glycogen is this complex molecule like this, made up of 1,4-glycosidic bonds and 1,6-glycosidic bonds. 1,6-glycosidic bonds are occurring at the branches, what we are calling the branches. Now, these ends that are linked by 1,4 are called non-reducing ends. Then this is referred to as the branching. Now, the first step would involve glycogen phosphorylase, and the work of glycogen phosphorylase is to break down 1,4-glycosidic bonds. And as it breaks them down, it's phosphorylating the glucose at carbon number 1, so it generates glucose 1-phosphate. I think that bit I had explained. So that's the first step. And the second step involves an enzyme called the debranching enzyme. Now, this debranching enzyme, sorry, it's bifunctional. It's a bifunctional enzyme, meaning it has 2 roles. And the first role, it performs this role as a glycosyl phosphortransferase, or what we also refer to as an oligo-alpha 1,4,1,4 glucan transferase. So what is happening is that these bonds, the 1,4 oligo bonds, are moving from the residues of one branch to the residues of another branch. That's the first step. Then the second step, the amylo alpha functionality of the debranching enzyme breaks down the 1,6-glycosidic bond. And in that case, that will be referred to as the alpha-1,6-glycosidase. So it breaks down the 1,6 bond at the branch so that then you have a single residue like that. So I think this is the image that can explain what is happening here in a proper manner. So the 4,4 bit, the 4,4 transferase activity, you're having at this branch, we said you're left with 4 residues on either side of the branch. So these 3 or 4 residues, the 1,4 bonds are moving to the next branch like this. Okay, so that is what we are referring to as the 1,4 glucan transferase activity, or what we also refer to as glycosyl transferase, glycosyl 4,4 transferase activity. Also calling it an oligo 1,4 glucan transferase. So this enzyme is called this way. It's also called 1,4 glycosylase, sorry. So it has several names. You can call it glycosyl 4,4 transferase. You can call it alpha-1,4 glucan transferase, or you can also call it alpha-1,4 transglycosylase. So it's just to move the 1,4 from this group of residues to these other residues, like that. Now, when that happens, you see now we have this molecule here. Okay, we have this molecule here that is straight, and then we have this branch. Okay, we have this branch. So this branch, this will be dealt with by an amylo 1,6 glucosidase that breaks it, and so then we are left with a glucose residue there. Okay. So this is exactly what I've been explaining still further. Okay. And the third step will be the formation of glucose 6-phosphate and glucose. So as you've seen, we have 2 enzymes, technically 3 enzymes. We have the phosphorylase, glycogen phosphorylase, and we have the debranching enzyme. And the debranching enzyme is bifunctional. And that's why we are saying we have 2 enzymes, but technically 3 enzymes, because the debranching has 2 activities. Now, our glycogen phosphorylase will give us glucose 1-phosphate. Okay, but the debranching enzyme is then going to generate, first of all, linear. The first step will give us a linear with just 1 branch with a residue. Then the amyloglucosidase, 1,6 glucosidase, will break free so that we get glucose here. So debranching ultimately will give us glucose. But glycogen phosphorylase will give us glucose 1-phosphate. And usually the ratio is 8:1. Okay, it's 8:1. So for every activity of glycogen phosphorylase, we're going to get 8 glucose 1,6-phosphate and 1 glucose for the debranching. Now, ultimately, of course, we're interested in glucose. That is what we want. We don't want glucose 1-phosphate. So the next step technically will be to convert glucose 1-phosphate to glucose 6-phosphate. Glucose 1-phosphate is converted to glucose 6-phosphate. And this is done by phosphoglucomutase. This is done by phosphoglucomutase. And then in the liver, glucose 6-phosphate is hydrolyzed to form glucose. So then that ultimately gives us the glucose that we want. And that would be, in a nutshell, what glycogenolysis is all about. We are getting glucose from glycogen with the help of quite a number of enzymes. But ultimately, this is brought down to glucose when glucose 6-phosphate gets to the liver, and then it's hydrolyzed to form glucose as it is. So this is what is happening in the liver now. So glucose 1-phosphate, with the help of glucose phosphoglucomutase, you get this glucose 6-phosphate. Then glucose 6-phosphate, with the action of glucose 6-phosphatase in the liver, and of course this enzyme is only found in the liver. Remember, in gluconeogenesis, this glucose 6-phosphate is just found in the liver. That's why that process happens in the liver again, mainly, and of course the kidneys, but this is mainly found in the liver. So this can convert glucose 6-phosphate to glucose. So yeah, just to emphasize that in the liver, the fate of glucose 6-phosphate, so in the liver and the kidney and intestines, that is where we have glucose 6-phosphate. But you see that mainly this is present in the liver, much as we saw that glucose 6-phosphatase is present in the kidney, and we're also adding here that it's present in the intestine. So which means all this can be utilized in producing glucose from glucose 6-phosphate. But the enzyme glucose 6-phosphatase is absent in the muscles, it's also absent in the brains. And as you see, these two organs require glucose. They really require glucose. So again, they have to depend on the liver, kidney, and intestines for the glucose 6-phosphate to be converted to glucose with the help of glucose 6-phosphatase. So then the muscles and the brain can be able to pick up this. So also not just that glycogenolysis is inhibited by fructose 1-phosphate at the level of the phosphorylase. So it is inhibited by this particular molecule. And so, of course, it can be regulated, though my aim is not so much to look at the regulation. My main aim is for us to be able to see how glycogen is broken down to glycolysis. So for your own reading, you'll be able to understand, or rather, anyway, simple and self-explanatory. You'll see how allosteric regulation is done. I just added this for the sake of you knowing. Also see how hormones, glucagon, epinephrine, and insulin are able then to regulate glycogenolysis. And of course, the significance of glycogenolysis. We know that the important thing about glycogenolysis is for it to be able to give us glucose, which we can utilize for energy production. So that is glycogenolysis for you. At this point, I would welcome any questions, any clarifications in as far as glycogenolysis is concerned.